Starting with the NTRK gene, and then we'll compare between the two trials and what's available data for comparison. Finally, testing. I know there is session for testing, so it's going to be only one slide in which tumor we should start to practice testing NTRK. So usually for molecular diagnostic, the way we started the whole practice in cancer is to do diagnosis under microscope or labs by tumor markers. Immunohistochemistry then came to the market, FISH diagnostic, and finally the molecular diagnostic for DNA and RNA. We have the fusion between two genes, gene amplification, mutations, and structural rearrangement. Now, fusions is documented in many genes, and it started long time ago, in around 1950s. The first NTRK gene was detected in the early 80s, and it took them long time till 2000, actually 17, and actually it's 1817 that we had 2 drugs approved in the market. So if we look at the landscape of kinase fusions in the whole solid tumor, fusions can occur in ALK, BRAF, and all these, and also the NTRK, which is the the hallmark. It's between any of the cancer. The one in the box in the reds, it's for NTRK 1 and 2 and 3. And if you look at the numbers, it doesn't mean you don't look at the colors. Colors just represent the novelty and which one was old and which one is they discovered it later. But if I look at the numbers, the higher number means the higher fusion, and thyroid cancer had higher fusion in the NTRK 1, 2, and 3, and mostly cumulatively higher fusion in most of the other genes like ALK and BRAF. For glioblastoma, also the same, and lung cancer, they are the most and higher fusion that present on the cancer. This graph presents that the frequency of patients with a higher fusion. This article was published in 2015, and it is the standard actually reference for our fusion incidences within different solid tumors. Now the NTRK represent the neutrophil tropomyosin receptor tyrosine kinase, and if we remember the whole tyrosine kinase tree, we only know in a very small part of the tyrosine kinase, and this is part of the trees. It's a neutrophil tropomyosin receptor tyrosine kinase, and it has 1, 2, and 3. And it's simply, if you look at the normal NTRK gene, what happened, it's attached and it's fused with another gene. It's not even related. It doesn't matter whether it's this gene, any gene. It could fuse with any gene, and it could present the NTRK gene fusion. And this has happened during dimerization when the LBD section of the NTRK gene is lost. Then simply the gene will be susceptible to fuse with any other gene that's available. NTRK gene and TRK receptors are expressed in neuronal tissue, so it's expected to maintain the health of CNS function, and usually within the fusion, the NTRK will confuse with any other gene. And usually the gene does not mutate or do amplification. It's only what happened, it's the fusion for the NTRK receptors and NTRK gene. Now, as I mentioned, that the first drug approved just couple 2-3 years ago, the Larotrectinib and Entrectinib. And those are the first generation, and the second generation is one coming to the market is very soon, and there is many trials also for other generation coming on the market. Now, frequency of TRK fusion within the body, it's very rare. And the first they discovered it actually in a very rare tumor. If you look at the left part, that the cancer enriched with the TRK fusion, more than 90% is the rare tumors, like the MASC, which is the salivary gland tumor, and the secretory breast cancer in adult, and pediatric secretory breast carcinoma, infantile fibrosarcoma, and cellular mixed congenital mesoplastic nephroma. This is why these drugs are tested in adult and pediatric in a very rare tumor, but then in the very early preclinical even stages. And then they found it in a lower preference, less than 5% with other tumor, which is the common ones. But if you look at the blue one, which is not bad percentage at all, which is 5 to 25% in thyroid, chest, and also lung. Even though it seems very small, this percentage is very critical in the treatment of solid tumor. Because if you look at chest, for example, it says, I have it highlighted here, pan negative, which is the same. Once the chest is pan negative, you expect TRK fusion and overexpression, and patient may be respond to these drugs. It's the same with the lung cancer. We'll talk about it right now, but just remember that it's very common in a rare tumor, and it's very uncommon in the other common tumors. So this is the classical pathway, the gamma PLC pathway, MAPK pathway, which is the RAS-RAF, and also the mTOR, which is AKT, PI3K, and mTOR pathway. Actually, TRK fusion, it's when it's stimulated, it binds to the BDNF, and then it activates the receptors, and it will be also inhibit the 3 pathway, which is the intracellular cascade, which is further will inhibit the cancer. So the first one came to the market is the microsatellite instability, and this is the second one. And actually we call it second tissue agonistic approval, because it's not, although it targets, but its work and non-specific in the tissue. It's not specifically in the tissue. So we call it Larotrectinib, and is the second, and Entrectinib is the third tissue agonistic approval. And the first one for sure is the microsatellite instability and DNA damage repair. So the first one came to the market is Larotrectinib, and the second one Entrectinib. They both approved in adult solid tumor and pediatric, and for patients who express Entrectinib gene fusion, and they are not enrolled for any active therapy. That's mean they're second, third, and fourth line. So it's either metastatic or surgical section is likely to result in severe morbidity and no satisfactory result. So it's almost like when patients run out of the options. Although again, we said if the patient does not harbor any tumor molecular targets, then they are eligible for TRK fusion inhibitors. And I will be clear with these things under testing. Now, will give you an example when to test and what tumor will be treated with these 2 drugs, which is coming soon. So Larotrectinib is highly selective. It inhibits the TRK fusion inhibitors, and it's tested in 3 early phases trial, and it's approved from these trials. Entrectinib is less specific, though it's inhibit the TRK and the ROS and ULK receptors. So it's highly suggested even from the preclinical trial to be used in the lung cancer. And it was tested in 3 adult trials and 1 pediatric trial. Now, starting with the clinical data with Larotrectinib, Larotrectinib, I'm sorry, and then Entrectinib. Each one I will present it separately, and then I will present a merged slide that it was presented in ESMO. I found it very useful. So patients in early phases, adults and pediatric, 3 trials, small number of patients, total number is 122 patients. The primary endpoint is based objective response, and its duration of response and progression-free survival and finally safety. Now the TRK fusion, it was required for this study. Dosing was 100 oral BID continuously, and for pediatric is 100 milligram per meter square. So this is the results. If you look at this, all the minuses mean the shrinkage in the tumor size. So look at the shrinkage of the tumor size. The overall response is around 81%, which is very high. It's an excellent response on these patients where we hope that there is no more response that it can be seen in this stage. And if you look around the cancer, thyroid cancer, colon, melanoma, soft tissue, lung tumor, just remember again, some of the cancers have higher percentage, but even with a lower percentage incidence, they still, if they are harboring Entrectinib gene fusion, that they will be respond from this graph. Now for the outcome and over treatment duration, the median duration of response was not reached. And it's very easy to tell from the first median time response, 1.8 months, and the assessment, most of the patients have a complete response, and the red dots are partial response, I'm sorry, and the red dot are the complete response. But then if you look at the median duration of 9 at 12, which is 1 year follow-up, still there is a huge number of patients that they still on active treatment and they still having response for a longer duration. And this will also suggest that progression is not soon with this medication. Now, again, the most common tumor, salivary, soft tissue sarcoma, infantile, and this is the same data. Adverse events were predominantly grade 1, which is very interesting, and no adverse event of grade 3 and 4. So it is really well-tolerated medication. The most common adverse effects seen, more than 20%, were fatigue, nausea, dizziness, vomiting, cough, increase in ALT, constipation, diarrhea. I want to also highlight again that those drugs can produce CNS side effects because of their neuronal mechanism of action, because Entrectinib gene itself exists on the neuronal tissue. And there was no treatment-related deaths. With the second drug, Entrectinib, it's almost the same design, early phases, primary endpoint with the overall response and duration, and the secondary is the progression-free survival. Doses is 600 every day for 28 days, so it's a once daily regimen. Now, this is the adult trial, and there is one trial, but I'm including the adult trial, 54 adult patients with Entrectinib fusion positive, and also 355 patients overall will considered all for safety. Now, if you look at this trial, because from the mechanism of action and the big glycoprotein engagement of Entrectinib, I just want to emphasize that big glycoprotein substrate, it's very common in tyrosine kinase, all the small molecule tyrosine kinase. So this is why they have to study the big glycoprotein. If it's highly, if it's less substrate, so it means it could work in the brain metastasis, and we'll talk about that. So there was a high percentage of patients with non-small cell lung cancer, around 20%. And if you look at the efficacy and the response, it's almost similar graph from the Larotrectinib. It was very effective, and the response was around 57, so it's lower percentage, with 7.4 complete response and 50% partial response. And those are the distribution of cancers: lung, salivary gland, breast, thyroid, and colorectal. But the median duration was only 10 months, while in Larotrectinib was not reached after even 1 year. So the side effect was more than 20% fatigue, constipation, dizziness, weight increase, cognitive impairment, again because of CNS effects, vomiting, cough, arthralgia, but most commonly reported side effect related to cognitive disorder, and there was no deaths related to the treatment. The next one is the median duration of response. Again, if you look at those patients in 1 year, there is still a patient responding, and most of the patient is still active on treatment. That means they did not progress, and it even with Entrectinib. Now they looked at the patient with CNS metastasis. Again, as I mentioned, that it was from the mechanism of action of Entrectinib. It was proposed that it crosses blood-brain barrier. So the efficacy, the pink one is from with the CNS metastasis as baseline, and the blue one, no CNS metastasis as baseline, and all these patient groups were responded to treatment. They did not include CNS metastasis in Larotrectinib. It was less than 5% of the patients, so they didn't analyze this group because, again, it was not proposed that it crosses blood-brain barrier such as Entrectinib. So definitely the great efficacy was seen in patients with or without CNS metastasis, and those even group of patients had a complete response while they have a CNS metastasis. I think this is very crucial and important, especially in lung cancer, as we mentioned, because around 50% of the non-small cell lung cancer patients develop brain metastasis. Now, if we want to combine the two trials just for summary, we have here the overall response with Larotrectinib, 80%, while Entrectinib, 57%. The median duration of response was not reached in Larotrectinib, and it was only 10 months, which is, I think, an excellent number still for the solid tumor, with a progression-free survival of 11 months and not reached on the Larotrectinib group. And this one for the time duration of response, again, this is the duration, 1.8 months. You can see that it's much longer response with Larotrectinib versus Entrectinib. Now this is a survival modeling, or what we call it in economic survival extrapolation. They take these survival and progression-free survival models from the original trials, and they try to see in a lifetime horizon, which means not in 10 years, not in 20 years. They see forever when all these patients will die. Now, if the graph did not overlap with the original graph, it means that there's still patients are alive after a long time, which is more than 20 years, which proposes curative effect. Now, if you look at the Larotrectinib survival, it was in 10 years, still there is around 40%. In 10 years, 40% are alive, which is amazing number. And then at 20 years, it goes lower, but it does not touch the original graph. It means There's still patients alive after a long time. It's definitely not the same scenario, and was a sharp decrease in survival curve with Entrectinib. So it gives us a hint that Larotrectinib was higher in survival than Entrectinib. Of course, these models, it's classical. We use it in economic, and they used it in Crizotinib, and after a long time follow-up, it matches their survival extrapolation. Quickly, interaction, if you look at the blood-brain barrier, big glycoprotein work as a guard, which doesn't allow drugs that goes to the brain. This is a trial used in a novel vitro tissue model, and they found that Larotrectinib has, if you look here, the red part is a strong substrate, and here a weak substrate. So Entrectinib has a weaker substrate for big glycoprotein. That means it crosses more the blood-brain barrier. This is why from the vitro trials, they suggested that Entrectinib can be active in the CNS metastasis, not like Larotrectinib. The last thing in the testing, the TRK fusion can be screened by immunohistochemistry, FISH, and next generation sequences. When should we test? It depends on the tumor type, although it is active across most of the solid tumor. Patients have curable options. If the patient have curable option, we should not test. If they don't have, then we have test. Now, this is not a standard guideline, and more it depends on the practice, and next generation sequences is the standard of practice in many other places around the world, even at King Faisal. But the problem is that it's a little bit expensive. So I'll give you example in papillary thyroid cancer. If the patient is BRAF mutated, is 50%, then you don't have to do all the next generation sequencing. What you should do, if they are not BRAF mutated, then you screen for Entrectinib by immunohistochemistry, which is more cost effective than doing the next generation sequencing. Now, from the beginning, it's recommended to do it because they have lots of genes involved. So it's good to do the next generation from the beginning, to do everything together, EGFR, RAS, ALK, and the Entrectinib. Just mutation, they have multiple mutation. If they are positive for these mutation, you don't have to test for Entrectinib. If they are negative, definitely you don't have to waste money in next generation, and you can do immunohistochemistry. I'm sure there is many things. Just summary that all these, the two drugs are generally well tolerated, and an Entrectinib fusion oncogenic driver, a wide variety alteration in multiple tumor. All tumors, this is the recent recommendation, without non-oncogenic should be tested and screened for Entrectinib. Thank you very much.